Chairs and dear guests, before I start, I have to say that I'm really thankful for the opportunity to organizing a scientific committee, and I'm really honored to be here. In the beginning, I'm going to give you some information about mitochondria, a little bit information. So mitochondrium, whose major function is energy production, is a crucial organelle for high energy demanded tissues such as brain. In mitochondria, ATP is produced through oxidative phosphorylation system, which consists of 5 multi-subunit complexes. Mitochondrial DNA is a circular double-stranded molecule, and it encodes 13 mitochondrial proteins, which are the subunits of the oxphos complexes. Each complex contains at least one mtDNA encoded subunit, as you can see here, and several nuclear DNA encoded subunits, except complex 2, because all components of the complex 2 are encoded by nuclear DNA. Mitochondrial dysfunction is characterized by the disturbances in oxphos and ATP production. Oxidative damage that is caused by reactive oxygen species, which are also produced in mitochondria during ATP production, leads mitochondrial dysfunction and mtDNA mutations. mtDNA is more vulnerable to damage since it lacks protective histone proteins and has limited repair mechanisms compared to nuclear DNA. Mitochondrial dysfunctions and mtDNA mutations may cause many diseases, yet the brain appears most vulnerable to mitochondrial defects because it's the most energy-demanded tissue in the brain. Mitochondrial dysfunctions are suggested to be related with neurodegenerative disorders such as Alzheimer's disease and Parkinson's disease. The studies on postmortem brain tissues of normal elderly subjects and PD patients have demonstrated high number of mutations and deletions in mtDNA of substantia nigra neurons, which are the most affected ones in Parkinson's disease. Since our study is focused on Parkinson's disease, I'm giving some information about it. Especially complex 1 and complex 4 based mitochondrial dysfunctions are suggested to be related with Parkinson's disease development. The information about the vitamin D, the information about the relationship between vitamin D and mitochondria is very limited, yet we know that the enzymes involved in vitamin D metabolism are located in mitochondria. And also in 2013, the localization of the traditional vitamin D receptor, which is called VDR, in mitochondria was also shown. Although the mitochondrial functions of other steroid receptors such as glucocorticoid and estrogen receptors were investigated, there were no studies conducted on the mitochondrial effects, mitochondrial functions of vitamin D receptors until now. The data that show that vitamin D metabolism-related enzymes are located in mitochondria, and also VDR is located in mitochondria. And proteins that normally have functions in nucleus by interacting with vitamin D have also functions in mitochondria. Vitamin D deficiency is related with neurodegenerative disorders in many populations, and mitochondrial dysfunction is related with neurodegenerative disorders development. Have arisen a question: does vitamin D have a role in mitochondria, and especially in the development of neurodegenerative disorders? Given that in this study we aim to investigate whether or not the mitochondrial dysfunction, which is the major basis behind the development of neurodegenerative disorders, is related with vitamin D deficiency in neurodegenerative disorders, and the mitochondrial functions are affected by vitamin D deficiency or the disruptions in the vitamin D-related pathways. In order to achieve this goal, we determined the effects of vitamin D and its receptors on the expression of mitochondrial genes that encode oxphos subunits in primary neuron culture and in samples of patients with a neurodegenerative disorder. For our first aim, we collected blood samples of 101 Parkinson's disease patients and 44 cognitively healthy patients, healthy subjects. The Parkinson's disease was chosen as a neurodegenerative disorder model because mitochondrial dysfunction is one of the major bases behind the Parkinson's disease type pathology and has the lowest vitamin D level among the other neurodegenerative disorders in our population, and its clinical diagnosis and classification are more distinct among the other neurodegenerative disorders. So we measured serum 20-OHD levels and intercellular ATP levels of leukocytes. And we grouped the patients and healthy subjects according to their vitamin D levels. And we formed 3 groups, individuals with whose vitamin D levels lower than 30 nanograms, lower than 20 nanograms per milliliter, between 20 and 30, and higher than 30 nanograms per milliliter. So for our second aim, we performed primary cortical neuron culture from rat embryos. And for this in vitro experiment, we performed 2 independent set of experiments. In the first set of experiments, we treated the matured neurons with different concentrations of vitamin D. And for the second experiments, we silenced vitamin D receptors by treating the neurons with sRNAs. By meaning silencing the receptors, we silenced the traditional receptor VDR and the other suggested receptor PDA3, and we silenced both of them at the same time. Then we isolate RNA from both primary neuron culture and blood samples of PD patients and healthy subjects, and we detected mRNA expression levels of these 13 mtDNA encoded genes by qRT-PCR. Our results on vitamin D levels have showed that Parkinson's disease had lower vitamin D levels compared to healthy subjects. And when we compare the mRNA expression levels of healthy subjects according to their vitamin D levels, our preliminary results showed that there was no difference. Yet the ATP levels in individuals whose vitamin D levels were higher than 30 nanograms per milliliter had increased ATP levels, and also the ATP levels and the serum 25 OHD levels were positively correlated. So this indicated that when vitamin D increased, the ATP levels also increased. And when we compare the mRNA expression levels of PD patients, again according to their vitamin D levels, our preliminary results showed that in patients whose vitamin D levels higher than 30 nanograms per milliliter, MT-ND4L from complex 1, MT-CO1, CO2 from complex 4, and ATP6 from complex 5 were upregulated. And again, in these patients, the ATP level were also increased. And the, again, ATP levels and serum vitamin D levels were positively correlated. This also indicated that when vitamin D levels increased, ATP levels also increased, same as healthy patients, healthy subjects, sorry. So in cell-based in vitro experiments, when we treated the neurons with vitamin D, different concentrations of vitamin D, we had different effects on mRNA expression levels. But in here, when we treat the matured neurons with 10 to minus 8 molars of vitamin D, the physiological concentrations, most of the complex 1 genes were upregulated while the other ones were downregulated. But in these neurons, the ATP level were higher. And when we silence the vitamin D receptors by sRNA treatment, we saw that most of the complex 1 genes, again, except ND1, and all of the other genes that are subunits of the other complexes were downregulated. These results were in harmony with the vitamin D treatment and the clinical data. If I summarize our results on mRNA expression levels, in Parkinson's disease patients whose vitamin D level were higher than 30 nanograms per milliliter, MT-ND4L from complex 1, MT-CO1 and CO2 from complex 4, and ATP6 from complex 5 were upregulated. And in vitro results, when we look at the in vitro results, when we treat the neurons with vitamin D, again, we saw that the MT-ND4L from complex 1 were upregulated. And when we silence the vitamin D receptors in these neurons, in matured neurons, we again saw that NDL4, CO1, CO2, and ATP6 levels were downregulated. And if I summarize our results on ATP levels, both healthy subjects and PD patients whose vitamin D levels were higher than 30 nanograms had increased ATP levels, and the ATP levels and serum vitamin D levels were positively correlated. And in vitro assays, neurons treated with vitamin D had higher ATP levels. So this again indicates that when vitamin D levels increase, ATP levels are also increased. So we assume that when vitamin D might be a molecule that can boost energy production. In healthy subjects, we solved no difference between mRNA expression levels, yet we saw that ATP levels were higher. So this mechanism probably might work not through mtDNA, but through nuclear DNA encoded mitochondrial genes, yet it's not shown yet. And we also know that one of the major mechanisms involved in PD pathogenesis is mitochondrial dysfunction. So our results showed that higher levels of vitamin D might try to compensate the dysfunction by increasing the expression of certain mtDNA encoded genes and ATP productions. These genes were subunits of complex 1, complex 4, and complex 5. And it is important to note that these genes, especially complex 1 and complex 4, are also suggested to be related in Parkinson's disease development. So this is our team, our group behind these works, and I'm really thankful for this opportunity for especially to Dr. Gezanak and Dr. Dursun, and thank you so much for your attention.
Thank you, Dr. Alailoglu. Again, due to time limitation, I can only allow for 1 or 2 comments or questions or any suggestions, ideas. Yeah, please, Dr. Karas.
Thank you very much for sharing your research with us. I saw in the graph you showed in the last few slides that this positive effect does not have a U-shaped relationship. I mean, as higher as 25-hydroxy goes, that means that you have an upregulation, an increase in ATP and the other OXPHOS genes. Is that correct?
May I have the question again? I couldn't understand.
The clinical message is that maybe regarding and based on your results, there is not a cutoff value beyond which vitamin D beneficial effects are implemented. I mean, you can get as higher in Parkinson's disease in order to have this boost in ATP, or there is a cutoff because based on your results there is not a cutoff.
Cutoff value for vitamin D or ATP? I couldn't understand that.
Did you use vitamin D or calcitriol?
For the neurons?
Yes.
Cholecalcitriol. We use that.
They used calcitriol, so you cannot.
Okay. Usually we have a beneficial effect up to a certain concentration of 25-hydroxy, and above that, in the current literature, that seems to be a U-shaped relationship, where you do not get a benefit. That seems not to be to comply with these results.
For in vitro results, you mean?
Yes.
In in vitro results, these were the preliminary results and the treatments, the results for the treatments for 24 hours. So we treated these neurons for also 48 hours. And it's not in here yet, but in 48 hours we saw that in higher concentrations also the ATP levels were increased, not just the lowest one, actually.
Okay, if we don't have any other questions, thank you, Dr. Alailoglu. And let me invite the last